Thank you again, Professor Weisman. Well, we must continue now. So again, we have an honor or having an authority in psychiatric genetics, Professor David Pose. Professor Pose is at the Psychiatric Neurodevelopmental Genetics Unit at the Massachusetts General Hospital. Professor, thank you very much. It's an honor to have you here.
Well, thank you very much for the invitation and being a part of this really wonderful initiation of what I hope will be a very long-running institute. And I need to find this, okay. Just want to start by saying everything that I'm going to show you here is really the result of spending a year of a postdoc with Myrna Weisman, and she taught me everything I know about family studies. So, and I also at that time decided that I really, while I was interested in adult psychiatric disorders, I felt like I didn't want to be in competition with her. And so I decided that I would really focus more on neuropsychiatric disorders of kids, and it was a very opportune time in that I was invited to become a part of a family study on Tourette syndrome, and that has led to some of the things that you'll be seeing. I also have to say that I woke up in the middle of the night having sort of panic attacks because what I thought I was going to say was really not consistent with what had already been going on in the meeting, and so I completely reorganized my talk. So if it sounds a little sketchy and not terribly coherent, it's because it's been changed in the last couple hours. I also want to emphasize that I'm a bit of an outlier when it comes to psychiatric genetics because I spend a lot of time thinking about the phenotype. And in my opinion, I have to say that I have no financial relationships to disclose, primarily because I have no financial relationships. So I start by saying this: the three most critical aspects of genetic studies of complex disorders. The first one is phenotype, the second one is phenotype, and the third one is phenotype. If you don't know what the phenotype is, you can search for a lot of genes, but you will probably never be able to find an association between a gene, and if you don't have a phenotype, you'll be able to do some interesting things about that gene, but you don't really know. Eventually, you might find out what it causes. But I think you can shortcut, or I don't know shortcut, but at least you can hone in and make your studies more efficient by spending a lot of time thinking about what the phenotype might be. And the other thing I want to say, I just remind you all of the fact that the genetic code contains 4 letters, A, T, C, and G, and DSM are not in the genetic code. So DSM may have been really wonderful for allowing some research, like you just heard, to proceed, and it helped make it uniform across different sites. But DSM is not a great way of categorizing people for genetic studies. Albeit a lot of the stuff I'm going to talk with you about now has relied on DSM categorical diagnoses, but I will also show some things where those categories might be in fact falling apart. I know that you heard a very profound lecture yesterday on the genome-wide association studies from Professor Håkan and unfortunately, because I got confused about schedule, I missed the talk, so I'm not exactly sure what he said, and I hope I don't repeat too much. But what I'm wanting to show here is some results from analyses of all of the genome-wide studies that have been done for ADHD, and probably present the outcome of those results in a slightly different way, and then show some similarities to some other studies. These are the summary of at least the 5 studies that were done prior to 2009, and you have the obligatory slide where nobody can read it, but I'll just tell you that these are the top hits of 5 different studies, and if you could read it, what you would see is that there's basically no similarities between the hits. So not really any replication. However, a student of mine who is from a university in Nijmegen in the Netherlands took all of these data and developed a pathway showing or trying to integrate what these findings might be. And he took all of the genes that had achieved at least a 10 to the minus 3 association in these GWAS studies and plotted where those genes occur or where they function in the nucleus or in a neuron. And where you see the arrows indicates interactions between genes, and there are some missing arrows here, but that it indicates that all of the genes that were the genes that are in yellow were the ones that were in the top hits in the GWAS. The ones that are in green are genes that have been implicated in knockout studies in mice, and the ones that are coded in red are those that are sensitive to stimulants and in treating ADHD. I also call your attention to this gene, CDC42. I'll say a little bit more about it later. Also ERK1, and there's some others scattered around that I'll call your attention to again a little bit later. So instead of focusing on a specific gene, the work now that Gert Polman is doing is looking at these pathways to see if there are any hints as to how you might understand better what this pathway is that would then eventually lead to hopefully better intervention and treatment. These are the results of 5 GWAS, I'm sorry, I went backwards. These are the results of the genome-wide association studies, again done prior to 2009, 2010, and using the same strategy of taking all of the hits that were at least significant at 10 to the minus 3. Dr. Polman's categorized all of those genes into genes that were involved in neurologic disease, development of the CNS, development of neurites, etc., and then developed again a pathway this is in the extracellular matrix and cytoplasm of the nucleus in the developing neuron. And again, these are showing some interactions between genes, and here is the CDC42. Again, if you also look through, you will see, and I can't find it now, maybe it's in the next slide, that this is now in the developing connection, sorry, that here you have the CDC42 again, here's ERK1, and if you look carefully, you'll see some of the same genes that were shown in the pathways for ADHD. So what you're seeing here is that while ADHD and autism spectrum disorders, according to the DSM, are 2 different categorical disorders, what we're seeing at the genetic level is that there are underlying shared genes that are involved in nerve development, neurite development, and synapses. And the CDC42 is a very interesting one. It's called the cell division cycle 42. It's a GTP binding protein, and it binds in the active state, the GTP bound state. It is related to the regulation of signaling pathways that control cellular functions, including cell morphology, migration, endocytosis, and cell cycle progression, so that it's a very critical gene in the developing brain, and it appears to be very important for the development of both ADHD and autism spectrum disorders. And the interesting thing is that when you look through the GWAS results, it does not show up as one of the top hits, suggesting that it's probably a completely normal locus without any kind of mutational events or variation. But it is it's important because it's interacting with other loci that are potentially variable, and that potentially changes the function of CDC42 in a way that initiates a different cascade of brain development. I want to now go to some of the work that I've been involved with over the years, and as an illustration of how you begin to understand that so-called comorbidities may in fact be actual variable expression of some of the same underlying factors, etiologic factors that may be at work in families. Here's a summary of family studies of obsessive compulsive disorder that have been done since 1995. And I limit it there because all of these studies use essentially exactly the same methodology that Dr. Weissman just spoke about, ascertained through individuals affected with obsessive compulsive disorder, all families, all members of the families, at least all of those that were willing to participate, were directly interviewed. The best estimate procedure was used to make diagnoses in all of the interviewed, and we got family history information about all of the non-interviewed individuals. And then best estimates were made. And in fact, Bob King, who is a child psychiatrist at the Child Study Center, has been working on this and many of the other projects that I've been involved with, refers to the best estimate process as a pretty good estimate rather than the best one. It I think gives you a very good estimation of what it is, but because of the way that the data are collected and not all of the individuals are directly interviewed, it is probably not as accurate as a direct face-to-face clinical assessment. But what you see here is interesting. In the study that we published in 1995, we had 100 families, about 466 relatives, and found that OCD occurred in about 10% of the first-degree relatives, compared to, and I've missed something here, these are the controls here, so compared to about 2% of the relatives of controls, and controls were individuals who were screened for the presence of OCD, but were not excluded if they had any other psychiatric disorder with the exception of psychosis. In a study that was published 5 years later by Jerry Nestat at Hopkins, again, the same design. They had about 90 families, and again, interviewed all of them available and used the best estimate procedure, and in fact found a remarkable similarity in the recurrence risks of OCD. What we also learned in studying these families was that there were a lot of relatives, a number of them, who had significant obsessions and compulsions but didn't quite meet DSM criteria. And they may have had significant obsessions, compulsions, but didn't spend enough time during the day. You had to spend at least an hour a day to meet diagnostic criteria. So we classified these as subclinical obsessive compulsive disorder individuals, and Jerry Nestat's group referred to them as probable, but the criteria were essentially the same. And again, found really quite remarkably similar results. And then sometime later, 5 years later, 3 studies were published in which families were ascertained through children with obsessive compulsive disorder, and then all available relatives were interviewed. Nadia Shabain did her study in Paris and found that 17% of the first-degree relatives of children and adolescents with OCD met criteria for obsessive compulsive disorder. Greg Hanna at the University of Michigan published, again, a study of families ascertained through children, found that 22.5% of them met criteria, of the relatives met criteria, and Cesar Rosero de Campos published a study in which that we had been working together on and found that 22.7% of the relatives met criteria. The remarkable similarity of these two is, again, makes you feel more comfortable in these results. But pointing out here that when, and this is seen in other family studies of other psychiatric disorders, that early onset disorders appear to be more familial. Now, just because something is familial doesn't mean it's genetic. And so twin studies were done. Again, there were a whole slew of twin studies done since the early 1930s, I think. But most of them were case studies. And these studies represented here are ones that at least had enough twin pairs in them to make the results a bit more believable. Carey and Gottesman in 1981 were the first to publish a study that had a small number, but a relatively large, it was the largest study done to date. They looked at DSM-3 OCD, and then Bolton in 2007, using DSM-4 OCD, studied a remarkable 854 twin pairs, where at least one of them had a DSM diagnosis of OCD, and found estimated heritability of, and these were children, found an estimated heritability of about 47%, suggesting that 47 to 50% of the variability that you see in OCD, in childhood OCD, is due to some sort of genetic variability. Other studies done looking at obsessive compulsive symptoms, not a diagnostic category, also demonstrated very similar heritability estimates. Clifford in '84, Janelle in 2000, and these two here are from a population-based twin study in the Netherlands. Again, all showing, these are slightly lower here, but all showing estimates of heritability in the 0.4 to 0.5, maybe 0.6 range, suggesting that the familiality that we're seeing is indeed in part due to some underlying genetic mechanisms. And I should also point out that these were estimates from children. Twins are, studying children in the general population. I started initially in the area of childhood neuropsychiatric disorders, studying Tourette disorder, and we published a study in 1991, and subsequent to that, there were at least 8 or 9 additional family studies that were published for Tourette. And looking across all studies it's not really a meta-analysis, but looking at the results across all studies, suggests that the risk of Tourette disorder is about 10% in the relatives of people with Tourette. The risk of chronic tics, which is defined as either having motor or phonic tics, but not both. Actually, a diagnosis of Tourette requires both motor and phonic or vocal tics. The chronic tics require just one or the other, but not both. That risk was about 15%, and it was surprising that in our first study we found that the risk of obsessive compulsive disorder, I call it now here OCB because of the criteria used in other family studies, but in our study we used the DSM criteria for OCD, found that 12% of the relatives met criteria for OCD, and these people did not have any tics. And as far as we knew, we hadn't really paid that much attention to OCD in our ascertainment of probands, although we had assessed it. And there had been some descriptions of the fact that OCD tended to be more prevalent as a comorbid condition in Tourette's. And if anything, our hypothesis would be that this was a subset or a subtype of Tourette that included some obsessive compulsive symptomatology. So in order to examine that, we looked at our family data to look at the rates of Tourette disorder, chronic tics, and OCD, and found this quite interesting pattern. what's true about Tourette disorder is that in the general population, boys are 3 times as likely as girls to get a diagnosis of Tourette. They're about equally likely to get a diagnosis of chronic tics, but the prevalence of Tourette disorder is higher in males than it is in females. And for OCD, the prevalence of OCD varies depending upon the age of the population. But in this study, what we saw was that we saw this same preponderance of males who had Tourette disorder, equal frequencies basically of chronic tics, but we saw twice as many female relatives meeting criteria for obsessive compulsive disorder, suggesting that there might be some sort of sex-related expression with having the same underlying genetic factors might be expressed differently in males and females. But we still wanted to know whether this was a subtype of Tourette or whether the OCD that we were seeing in these families might actually represent a different expression of the same underlying factors. And what we saw when we separated families on the basis of the initial index case having OCD or not, we found that the rate of obsessive compulsive disorder was basically the same, and it was independent of whether the index case had OCD, which suggested to us that at least the OCD that occurred in these families probably was due to the same underlying risk factors that were increasing the risk for Tourette disorder. Interestingly, in the study that was published by Cesar Rosera Campos, we also, because of these findings in the Tourette families, began to look for tic disorder in the relatives of our childhood onset OCD, and found that, in fact, the rate of tic disorders in the relatives of children and with OCD was considerably higher, 10 times as high as our rate in the control sample. Again, suggesting that there is some sort of relationship. And although I don't have the slide, we did do the same analysis of looking at families of probans who had OCD and tics, comparing them to families where the proban had OCD without tics, and found that there was indeed a different relationship, that the tics occurred predominantly in those families where the proban had tics, suggesting again that there are at least 2 types of OCD, childhood onset OCD, one that appears to be related to tic disorder and one that does not. And if you look at the adult data, you also see the same kind of pattern. You don't see such an increase in tics in the relatives. You do see a slight increase, but when you separate on the basis of whether the proban had tics or not, the tics essentially only occur in the relatives of probans who had tics, again suggesting that there may in fact be an adult onset type of OCD that's related to Tourette, and an adult onset OCD that is not, and then there is yet another kind of OCD which is predominantly on adult onset that appears to be isolated cases. Again, twin studies suggest that at least this recurrence of Tourette is heritable. That the heritability of Tourette disorder is about 50 to 60%, a little higher than what was seen for OCD. Now, it's also been reported that there is a large comorbidity of, at least in clinical populations, of Tourette disorder and attention deficit hyperactivity disorder. And there was a lot of controversy back in the early 90s about whether ADHD was actually showing the same kind of relationship as obsessive compulsive disorder had been shown in these other family studies. So we looked at it and found that indeed ADHD was increased in the families, but when you separated on the basis of the proban having ADHD or not, you found that essentially all of the ADHD, or at least the familiality of ADHD, was really in these families where the proband had ADHD, and the rate of ADHD in the families where the proband did not was basically the same as the population, suggesting possibly that there is a subtype of Tourette disorder that is related to ADHD, but not in the same way as OCD. So we then did a study. We got another grant to basically do this study to look at this relationship between Tourette and ADHD, and looked at different kinds of families. Here, the proband had Tourette disorder only. We had 75 families there, 75 families where the proband had both. We also ascertained families, 40 families where the proband had only ADHD with no tics, and then about 50 families of controls. And what you see is that the rate of Tourette disorder was the same, the rate of tics was the same, and there was also an elevation of ADHD in these families. Surprising to us, there was an elevation of Tourette disorder in these families, and a very high recurrence risk of ADHD. What came out of this was surprising to us, in that the Tourette disorder that occurred in these families was basically occurring in families or in relatives who had both Tourette and ADHD. So there was an overrepresentation of this co-occurrence even in families of ADHD alone. And these numbers here as well, even in the Tourette-only families, you had this overrepresentation of comorbidity of Tourette and ADHD, and you saw that also in the families where the proband had both. These were all higher than you would expect by chance. All the bolded yellow entries indicate significantly higher than the controls. So you saw this very interesting phenomenon where you saw an overrepresentation of the comorbidity even in families where the proband had only ADHD. Well, here's a case where ascertainment bias can really be beneficial to you. Turns out that all of the ADHD folks had been seen at the Child Study Center, but they had presented with ADHD, but then because there was a Tourette clinic as well as an OCD clinic, they were fully evaluated for both. And we didn't know this at the time when we selected them, but about half of the probands with ADHD also had OCD. And that was very fortuitous to us, because it helped us really understand what was the underlying strategy, or what underlying cause for this overrepresentation of this comorbidity of Tourette and ADHD. I'll just go to the next. Well, the first thing we did was to look to see whether there was some relationship about the onset of ADHD compared to the onset of Tourette disorder. And what we saw was that if the onset of ADHD occurred after the onset of Tourette disorder, it looked like really this was an indication, or a situation in which the child was an unfortunate individual that had two independent disorders. If the onset of Tourette disorder was less than or equal to the age of ADHD, what you saw, I'm sorry, it should be the other way. If it was about the same or ADHD was less earlier onset, what you saw that it was, you saw this preponderance of the comorbidity. So that, so in this case it looked like there might be some etiologic relationship. In this case it looked like they were two independent disorders. So then, in subsequent analyses, and again, this is a very, very busy slide, but I just point out two cells in each of the slides that you see here. Here are families where the proband did not have ADHD, had Tourette disorder but not ADHD, and we looked at this comorbidity of Tourette and ADHD and saw that it really did not occur in these families, but it did occur when and this thing is dying, it did occur when the proband had OCD. So this was a proband who had Tourette disorder and obsessive compulsive disorder, but not OCD. But not ADHD. And you saw this preponderance of the comorbidity of ADHD and Tourette disorder. And it has stopped working. Next slide, please. If you looked at the families now where the proband had both Tourette and ADHD, you saw the same pattern, that you had, if the proband did not have OCD, the occurrence of Tourette and ADHD was not higher than you would expect by chance. But if the proband did have OCD, the occurrence of Tourette and ADHD was indeed higher than we expect by chance. Next slide, please. Now, looking at the ADHD, remember that these are all ADHD probands that did not have any tics. About half of them didn't have OCD and about half of them did, and you see exactly the same pattern where the co-occurrence of ADHD and Tourette disorder was indeed related to and predicted by OCD in the proband. And about the same time, a study came out, first author by Carol Matthews and Marcos Grados, in which they demonstrated that individuals that had Tourette, ADHD, and OCD appeared to be the most heritable type of Tourette disorder. And in fact, when we went back and looked at these relatives who had both Tourette and ADHD, essentially all of them had either were diagnosed with full DSM OCD or subclinical OCD or had significant obsessive compulsive behavior. So that this comorbidity that we thought was a comorbidity of Tourette and ADHD was really predicted very strongly by the presence of OCD in the proband. And so again, here we're seeing that the categorical things seem to be not really able to discriminate or tell you about what might actually be underlying the actual etiology. Next slide, please. So once you have this evidence of familiality, and that is genetic, you obviously then want to try to identify genes. And there are 3 different strategies that work. Linkage was initially used because that's the only strategy that we knew about early on. And it turns out that it works really well for rare conditions, but not so well for common conditions. So, yikes. So the next strategy then was developed after the DNA revolution, explosion, the genome project, and so on. And we're now able to do case control studies, and they work well if it's a common disorder with common underlying genes. And then another strategy, which you heard about as well yesterday, is looking at, next slide, copy number variants, which are very rare in the population, and usually have a larger effect. Next slide, please. 2 different strategies for case control or association studies. Next slide. And just talking a little bit about what genome-wide association studies are. The method is identical to a candidate gene studies, except now you're doing it in an agnostic way. You're not specifying that you believe any particular gene is involved. You're just going across the entire genome looking for associations. Next slide. Basically, this is what it looks like when you're scanning the genome with about 1 million SNPs. Next slide. There's a lot of challenges. Most of them are surmountable. The first one was computational. You're now dealing with more than an order of magnitude larger databases. It's several orders of magnitude more data, and so you need much, much better computing power, which fortunately was developing at about the same time that the genetic technology was developing. Next slide. Next slide. The problem with doing case control studies is that there are genetic differences between populations. And if you don't select your cases and controls appropriately, you can get spurious positive results, not because the genes or SNPs are related to the disorder, but because your cases and controls come from a different populations. Next slide. So in order to do that, you do multidimensional scaling analyses and principal components, and I'll show you what that means in an example here in the OCD GWAS that I'm about to talk about. This was done as part of the International Obsessive Compulsive Disorder Foundation Genetics Collaborative. It included initially about 2,500 affected individuals, which included 700 trios and about 7,000 controls. Next slide. 21 different sites contributed. Next slide. And after quality control, we were left with about 1,500 cases, 6,300 controls, and 400 trios, meaning that we had about 1,900 affected individuals in this sample. Next slide. And this is what we did before to ensure that our cases and controls were actually coming from the same populations. What we took was 50,000 SNPs and did the multi-dimensional scaling, and we saw this plot. Next slide. And we decided that this is the sample that we would analyze initially, and you see this sort of gradation, and what that represents is the upper part of the ellipse is individuals of European descent that come basically from Northern Europe, and the ones on the bottom are ones that come from Southern Europe. And these two little subsets on the right-hand side, the lower one are individuals who had 3 or more grandparents of Ashkenazi Jewish origin, and the one that's intermediate is individuals who had 2 or fewer grandparents, or 2 or 1 grandparents with Ashkenazi Jewish origin. We eventually analyzed these separately, all of these 3 groups, but then did a meta-analysis to put them together. Next slide. And these are the results. And again, rather than grouping them by strength of association, I grouped them by where they occurred in the genome. And you see that there are 3 SNPs on chromosome 6 that identify a gene called GRIC2, which is a glutamate receptor. Another 3 SNPs on chromosome 8 that are in a gene called ADCY8, which is a gene involved in immune response. Next slide. The highest hit was on chromosome 12, identifying a gene called FAIM2, and then chromosome 18, 2 SNPs in a gene called DLGAP1. I'll talk a little bit more about them. And then 4 SNPs on chromosome 14 that were associated with a gene called FUT2, and then finally the BTBD3, the one on the bottom, which I'm sure you can't read, was a gene that actually achieved genome-wide significance in our trios, and it's on chromosome 20, and interestingly, it's under the peak of a linkage study that was done on a very small OCD family, and this gene is right under that peak. Next slide. So I just want to talk a little bit about these genes. The BTBD3, it's a transcriptional, a gene that regulates transcription. It's expressed in the brain with the highest observed levels in childhood and adolescence. It's right when OCD onsets. Next slide. There are 2 genes that were very high in the association list in the trio sample that were correlated. The function was correlated with other genes that were very high in the case control sample. And I mentioned ADCY8. The ISM1 is highly correlated with that expression, and ISM1 has been associated with bipolar disorder and with fear memory, which has also been a strong area of study in OCD. It's also just point out that ISM1 was correlated with a lot of different glutamate-related genes, And there's been a lot of work looking at the glutamatergic role in the pathogenesis of OCD. Next slide. I'll just skip this one and go to the next one, and give you the results of the Tourette GWAS. It's again included about 1,500 cases of Tourette and about 7,000 controls. We did in fact have one genome-wide significant result, next slide, which is a gene called COL27A1. We didn't know what really to make of it. We still really don't, because it's a gene primarily expressed in cartilage. And to think of how cartilage might affect movement is, at least ticking movement, is a bit of a stretch. Although, looking at its expression pattern, it is expressed in the cerebellum during many stages of human development. So here's a gene that may have multiple functions that could in fact have some involvement in neurodevelopmental processes. Next slide, and next slide, next slide. So we're coming back to the phenotype. Next slide. Just want to give this one example of what we've been also doing with OCD. There are a number of different kinds of obsessions and compulsions that people have when they have OCD. Aggressive, contamination, sexual, hoarding, religious obsessions of symmetry, and somatic obsessions. And next slide. 6 different kinds of compulsions: cleaning, checking, repeating, counting, ordering, arranging, and hoarding. And next slide. A meta-analysis done by Michael Block from the Child Studies Center, looking at 22 studies, including over 5,000 people, showed that these categories of obsessions and compulsions can actually be reduced to 4 factors: 1 hoarding, 1 symmetry, called symmetry, 1 forbidden thoughts, and 1 cleaning. And the symmetry factor includes obsessions about symmetry, but then compulsions of ordering, repeating, counting. The forbidden thoughts is primarily obsessions of aggressive sexual or religious content and somatic, and then cleaning is obsessions of contamination and then behaviors. The interesting thing is that when you look at the relationship between these factors and the presence of tics, the symmetry factor is the one that occurs most often in those individuals who have OCD and tics. And if you look at the OCD that occurs in Tourette disorder, it is predominantly of the symmetry type. Next slide. We also did some analyses of the underlying genetic mechanisms using our GWAS data. We used the SNP data to estimate heritability, and also look at the kinds of genes that were identified, and it comes out that the Tourette disorder is more likely to be due to rare loci with larger effects, similar to the copy number variants. OCD is more likely to be due to more common loci with smaller effect, i.e. more like a polygenic. But the interesting thing is that the genetic correlation between Tourette and OCD is about 0.4. So again, individual specific genes probably related to specific symptomatology, but there is some correlation between the two. Next slide. And I think, well, just finish with this. In a study that was just published at the end of February, the Psychiatric Genomics Consortium looked over 5 different disorders to see if there were common genes that would be associated with each. And the findings were in the next slide, that there were 4 loci that surpassed the genome-wide significance. They were, and 2 of them were related to voltage-gated calcium channel subunits, and they were shown to be associated with all 5 of the disorders. So that thinking, and these seem, there's ADHD, autism, schizophrenia, bipolar, and major depression. So it suggests that clearly the brain is not categorized in DSM ways, that there are genes that may affect mental health, mental illness in a general sort of way, and then there may be other genes that in fact give more specificity to the symptomatology. And knowing this should eventually lead to better intervention strategies so that we can probably won't be able to cure, but we can do a lot to prevent and at least minimize the severity of these symptoms. And thank you very much.
Thank you, Professor Pols, for your excellent presentation.